Hello, everyone. I'm Hans-Peter Kiem, President of the American Society of Gene and Cell Therapy. Before we get started with our podcast, I wanted to take a moment to encourage all our listeners to consider registering for our annual ASGCT meeting. The annual meeting runs from May 16 to 20 at the Los Angeles Convention Center. But virtual attendance is available, and you can see every presentation from anywhere in the world. ASGCT's annual meeting is a truly fantastic place to learn from experts, connect with your peers, and share your science. The meeting covers everything from basic science to clinical trials, and more than 1,700 new pieces of research will be presented throughout the event in sessions and in our poster hall. You can learn more and register online at annualmeeting.asgct.org. Now let's get started with our podcast. Hello, and welcome to Giants of Gene Therapy. My guest today is another true giant in our field. He was the first to use the adeno-associated virus, or AAV, to deliver gene therapy to target sites in the body. Welcome, Dr. Flotte. Dr. Flotte is the Provost, Dean, and Celia and Isaac Haidak Professor at UMass Chan Medical School, with faculty appointments in the Department of Pediatrics and the Horae Gene Therapy Center. He's been at UMass since 2007. Dr. Flotte is also a practicing pediatric pulmonologist and a prolific researcher who has authored more than 280 manuscripts and been the principal investigator of multiple clinical gene therapy trials. He's also the editor-in-chief of Human Gene Therapy, the oldest gene therapy journal. He's currently investigating the use of gene therapy for genetic diseases that affect children, including alpha-1 antitrypsin deficiency and lysosomal storage diseases. Dr. Flotte has been an ASGCT member since the Society's beginning in 1996. He's served on the board and committees and spoken at the ASGCT annual meeting and other events. Again, welcome, Dr. Flotte, and let's get started from the beginning. Tell us a little bit about where you grew up and about your family.
Well, thank you so much, Hans-Peter. It's such an honor to be included in this podcast series, and I do appreciate your leadership of ASGCT. So, and I'm a little humbled to be telling you all of my story, but I will start at the beginning. So my family's from New Orleans. I was born and raised in Louisiana. My family's actually been in Louisiana for 8 generations. And we're not a very medical or scientific oriented family. But growing up, I would say I was both very intrigued with science, and I was also very much admired my own pediatrician, a guy by the name of Thomas Garvey. So I would say he was my first and most prominent physician role model as I grew up and went through school. And, my time in New Orleans, I stayed there. When I went to college, we didn't have a lot of money. So I went to the public university that was in New Orleans, a large public university, University of New Orleans. And then I went to medical school there at the public medical school, Louisiana State University, LSU. And so as I went through, my aspiration was generally to be a pediatrician. But when it came time to finish up medical school and go in for the match, I had done really well academically and thought I would go to see what it was about in some of the more prestigious academic institutions, which is how I landed at Johns Hopkins in the match.
Okay. Do you have any brothers and sisters?
Yeah, so I have 2 brothers and 2 sisters. I have a brother who's an engineer and a brother who is a lawyer. Which is what my father was. He passed away. My father passed away when I was young, but I think the default in our family was probably to be a lawyer. But at a certain point in time, I really felt like I wanted to have what I felt to be a more direct impact on human outcomes and on people, and also definitely was intrigued by science.
So when was that at that time? When, as far as you can remember, when do you think you kind of decided, okay, I'm going to do medical school?
Well, it's an interesting question. So actually, often great opportunities are born out of difficult circumstances. So actually, the thing that really made up my mind was my father had a sudden cardiac event, which he passed away from when I was a teenager, when I was 14. And it really created a sense in me that medicine had to do better so that we didn't have as many tragic outcomes for people. I just felt like this was something that we needed to all fight for, people to be healthier, live longer, and not have these kinds of tragedies befall families. So that was really a turning point. I was 14 at the time.
Wow. And then you decided to go to Johns Hopkins.
Yes, so that was another real turning point for me. So it was my top choice. I matched for residency at Johns Hopkins. I was inspired by really its alignment of clinical excellence with academic excellence, as some of our best medical schools and hospitals are. And I had determined to go into pediatrics, thought I might do a subspecialty, but I wasn't exactly sure what I would do. And the chair there at the time that I started was a guy by the name of Frank Oski, who was a prominent pediatric hematologist, who was a terrific teacher. And as I went along in my residency, I was exposed to many other physician scientists, including another LSU graduate who became chair actually at Hopkins, George Dover, who was also a pediatric hematologist. And he was a terrific example because he had an NIH-funded lab to study beta globin gene regulation. And then he would also see sickle cell disease patients in the clinic in the course of the same day. He might be in his lab, in his clinic, and I hung out with him. There were others like George who were really inspirations, and I started to see that one could be at the forefront of both science and medicine, and certainly the molecular biology revolution that was ongoing there that was in the 1980s. And it was, as I was a resident, that was really, things were really coming together at that point in time.
Mm-hmm. What made you go into pediatrics?
Well, pediatrics, as I said, it had been my original role model, had been my pediatrician.
Okay.
But I did consider a lot of other things. And in the end, the interaction with children and families ended up being something that I really resonated with. When I talk to my students now, to medical students, I tell them that the experiences in the clinical part of medical school are as much about finding out about yourself as they are about finding out about the specialties. And how does being immersed in one of these settings make you feel? I mean, does this feel like where you belong? And for want of a more precise description, it just seemed from the beginning that's where I belong.
Oh, this is so right, yeah. And then after Johns Hopkins, Terry, where did you go from there?
Well, I ended up being, so I evolved quite a bit over the time I was part of Johns Hopkins. So I started as a resident in 1986 and did my, residencies pretty flat out. But then it was before there was actually a fellowship match in pediatrics. I was basically hired as a pediatric pulmonary fellow before the end of my intern year. I knew I was going to do that. Jerry Laughlin was the division chief there. And he said, well, you have a potential for a career in academia, so you need to go and learn lab research. So when I started, actually at the very beginning, when I started my fellowship time in 1989, I participated in a program that the Cystic Fibrosis Foundation was funding, where I was able to go for 3 years as a special volunteer, quote unquote. In other words, I was paid as a Hopkins pulmonary fellow. But I was working in the lab of a basic virologist at NIH who turned out to be my most formative scientific mentor, Barry Carter, who later became a president of ASGCT, like yourself. And Barry is a wonderful, he's retired, he's a wonderful, curious scientist and also quite an individual. He's a New Zealander former rugby player who had such a joy of life and science. And the first day I walked into his lab, I was a resident, still planning my fellowship research project. He said to me, cystic fibrosis, that has something to do with the lungs and their sticky mucus, right? And we sat down then, and we had a little discussion about what I knew about CF and about chloride channels in the lung, and some of the molecular basis that was just coming together. We started the project before the CFTR gene was discovered. And then I'd done a little bit of background reading, about 8 or 10 papers of his on AAV. So I was like, this AAV is really interesting. So then that was my fellowship project, was to take AAV and vectorize it, if you will. And then when the CFTR gene came along, we put it into the vector, and that kind of got me off on that first AAV2 CFTR program, correction in cells, animal models. And by the time I finished my time at NIH, we were ready to do the animal experiments, which then led to going to treating the first patient when I was still an assistant professor at Hopkins. Barry had left NIH and gone to be the founding executive vice president at Targeted Genetics. And so Barry made the GMP vector, and we put it in the nose and lung of a 42-year-old woman with CF, and that was the first human use of AAV. And so here I was barely 3 years out from my fellowship, and I was the PI of the first human gene therapy trial with AAV.
Yep. I remember those days very well myself, when Barry moved to Seattle and Targeted Genetics. Those were very interesting days. So this was 1995, and you were the PI of that first study to deliver to use AAV for cystic fibrosis. Tell us a little bit about that. I mean, this was a very early start, and
it was a very early start. And of course, it was at a time when we had to go and defend the protocol. Every protocol had to be defended at the RAC, the NIH Recombinant DNA Advisory Committee. So it was some conference room up in building 1, I believe, near the cafeteria, where we would go and present our protocol. And I remember one of the biggest concerns was because we were putting AAV into the respiratory tract. One of the biggest concerns was environmental shedding. So we ended up to where we had to have the patient in the hospital for 4 days before the gene therapy, and then for something like 16 days afterward. So it was like a 3-week admission in isolation. And, I think everybody's got used to, all the clinicians got used to wearing PAPRs or N95s during COVID. We were having to wear the PAPR every time we went in the room. And what we now realize were homeopathic doses of AAV, because the manufacturing technology was not nearly as robust. It wasn't exactly homeopathic doses. We certainly were able to detect gene transfer particularly in the nose and in the lung. But it was there was a lot of hysteria about the possibility that some recombinant AAV could get out there and be spread around on the subway and cause some major problem. And I look back and I say, if only it was that easy to get AAV into cells, that was a real problem. But that was just a snapshot of what the time was like at the time. Right. I had been actually at NIH. Barry was actually on the rack when Steve Rosenberg did the first human gene transfer, which was the neo gene into the tail, the tumor infiltrating sites. And then shortly thereafter, Mike Blaze did the first ADA skit, all that at intramural NIH. So I was able, actually, I think he was not actually on the rack. He was on the NIH Institutional Biosafety Committee that hashed out a lot of that stuff that the rack heavily relied on for those first intramural studies. So it's a very interesting time where there was a lot of concerns that turned out not to be the most, we didn't know what the biggest potential risk benefits were, but that was there was a lot of focus on that end of it.
Well, then, of course, ASGCT was established just a year later, 1996, and I think you were one of the first members of ASGCT as well. I think this was all a very exciting time. I also remember that very fondly. Now then, though, I think we went through a little rougher stretch there in the late 90s, of course, with Jesse Gelsinger's death, and then also some of the other complications from patients with after stem cell gene therapy for immunodeficiency. Maybe now go back a little bit to that time, then late 90s now, how you remember that time.
Well, certainly I was thrilled when ASGCT came together because I, like yourself, had been trained as a gene therapy scientist, and there really wasn't an academic home for gene therapy scientists. I mean, I present data at the pulmonary meetings, and a lot of people were interested, but it was a more narrow audience, and so there wasn't as much of an opportunity to meet with other colleagues who were pushing viral vectors or non-viral vectors or other technologies forward. Interestingly, there was a little mini wave of interest with, some investment in biotechnology. In retrospect, not as big as what we experienced in a later phase. But then, yes, as you say, things started turning to the negative at a certain point in time with both the Gelsinger OTC, adenovirus OTC vector, and then with some of the insertional mutagenesis in the ex vivo gamma retrovirus trials in Europe. And, I think there were a couple of things that this taught us. I mean, the first thing, particularly with the adenovirus, was that we did not have great animal models to predict exactly where the safe dose range would be. We also were pushing, we hit the limit first with dose-related toxicity to adenovirus, but we couldn't really get to the right dose range to see dose-limiting toxicities with in vivo gene therapy until we got the manufacturing better. Then we started to hit these limits. The ex vivo retrovirus gene therapy trials also were great learning experiences because the improved understanding about vector technology and what the mechanisms of insertional activation were led to better designed self-inactivating LTRs and the like, which other people are more qualified to talk about than I am. But I think it just showed that these types of toxicities couldn't be fully predicted from any preclinical model. So by necessity, while we're not happy as that individual patients manifested the outcomes of these risks, it actually demonstrates the iterative nature of a field like this. You really have to get some data in patients to understand what the nature of toxicities are. And of course, this has come to the fore more recently with AAV, where we're hitting dose limiting toxicities of various kinds.
I was also impressed back then, yes, we had these deaths and these severe side effects from the gene therapy, but it's also showed, I think, the strength of ASGCT to go back to the basic science, to the bench and figure out what the problem was and come up with solutions to get into the next phase of clinical trials. I was actually very impressed how we as a society were all coming together and identifying the problems and then coming up with solutions.
Well, I mean, your contributions in that time were really critical, and certainly identifying exactly what happened with some of the leukemia syndromes from insertional activation. But you're right. I think it showed that this was not a set of technologies that was going to be handed off to drug companies into the clinic and then just go on its merry way. It required continual refining and learning from the scientific community in order to inform what the next generations of therapy were going to have to look like.
So now, though, since then, AAV has been very successful. Yes, there have been some side effects, but want you to talk a little bit about really the successes of AAV again, especially over the past, almost 10 years now. Glybera may not have been the biggest success. There were other issues. But then, we had other drugs that were approved here in the United States, and maybe, talk a little bit about that.
Well, thanks so much. Yeah, AAV, I'm certainly, because we were involved in the very early clinical translation of AAV, I do, have this rooting interest in AAV. And I think, interestingly, one of the things we discovered that really drove home about AAV from the earliest clinical trials was that it was an episomal vector. Now, that's actually in the textbooks and all that now. But at the time, the interest in AAV as a virus was because it integrates site-specifically in certain cell lines. It actually doesn't really do that in nature either, but at the time, people thought that it did. But when we started using it in a proliferating epithelium in the lung, it became quite obvious, and we actually showed this in primates early on, that it was episomal. And the logical thought, you'll go back to when most of the field was working with retroviruses. The prior thought had been, you're only going to get gene transfer in dividing cells, because for gamma retroviruses to get access to the nucleus, the cells have to go through mitosis. That turned out that was not the case for AAV. And because proliferating cells lost the episomal AAV, that whole thought process shifted the hammer of AAV gene therapy toward the nail of non-dividing cells, right? In other words, we didn't really, while there was a lot of theoretical thing about, dividing cells would be better, it turned out non-dividing cells were, and the most accessible target of non-dividing cells really was the retina. And so hence the program that resulted in Spark Therapeutics and CHOP developing Luxturna for RPE65 deficiency, which is in fact itself the optimal retinal disorder, because many retinal disorders cause death of photoreceptors, but that is a defect in retinoid recycling. And so photoreceptors are still there, but not able to function, particularly in low light vision. This is all history now, but at the time I was actually collaborating closely with Sam Jacobson and Bill Hausworth and trying to use some of what we knew about the safety profiles of AAV to help move forward. And at one point, Bill was collaborating with Gene and others on the correction of the RP65 dog and all that. So, some of our colleagues in Florida had a lot of important work there. But the point is that the field was persisting, going where the vector might work. And there was another little caveat to this. There was a point in time in the 90s when people were telling me drug companies, biopharma companies will never go after rare genetic diseases. These things have to be used in common diseases. Well, it turns out that putting a gene in works best for a single gene defect. Go figure. It's not the only thing it works for, but so when you have a retinal problem that is an autosomal recessive single gene disorder, it turned out it was the perfect fit for AAV. And so in retrospect, I think the Luxturna approval, which was the first one in the US, and Glybera had already worked for LPL deficiency in Europe with intramuscular gene transfer. But anyway, the Spark Luxturna product makes sense that it would work. And now AAV, with that spurred on by that AAV's ability to transduce neurons, also non-dividing cells, and with the ones in the lower neurons being most accessible, you see spinal muscular atrophy, Zolgensma, being approved. The interesting thing about the AAV9 SMN1 compared to the original, the Luxturna, the AAV2 RPE65, is that while it was certainly dramatic to see patients who were visually impaired regaining vision, to see babies with SMA1 be able to literally stand up and walk was transformative. I think we knew at that point there was no going back. AAV was going to end up being a massively impactful platform for certain genetic conditions.
Especially Zolgensma now. This is in many, many countries being used, correct? How many patients have been treated so far?
I think it's a couple of thousand patients,
exactly,
roughly.
Yeah.
I don't have the exact number.
Yeah. So this is,
Someone told me 2,000 patients altogether.
Yeah. And then most recently for hemophilia B. So I think there's been AAV has again been very, very successful.
Yes, indeed. AAV been approved now for hemophilia B, and there's also hemophilia A product approved in Europe, and more coming along all the time. And so the AAV approvals in particular have been extremely gratifying to see.
So where do you see that field with AAV going, and what are the remaining obstacles you think? I mean, what's of next for AAV, and again, where do you see the obstacles? And maybe include your own research there too.
Yeah. So, well, thank you for that. Well, I think that when we look at where AAV has gone, I think for treating small populations of cells, it's going to be really the current platform is going to work fine. For instance, in the retina and maybe lower motor neurons in the spinal cord. Where we're running into trouble is we're having to push the doses really quite high in order to get distribution to larger targets, like the entire musculature, for instance, in muscular dystrophy. Although it can be done, it is at a point where there is measurable levels of toxicity there. And same thing for global brain delivery. So we're at a point where we're trying to optimize getting the payload to the right cells when the individual disease process calls for it going to targets that have a larger total mass. So that challenges, on the one hand, manufacturing, scale up of manufacturing, but on the other hand, I think it really is a point where we need to work to improve potency of the vectors, meaning maybe the vectors have an enhanced tropism, or for some other reason, we can decrease the total protein load being delivered for the amount of effective transducing viral particles. That could be important, especially for conditions like muscular dystrophies. I'm sure we're going to have some muscular dystrophies where the current technology is approvable. We're seeing evidence of efficacy, but it could be safer. It could be easier. And I think that's definitely an issue. And then for global delivery to central nervous system, that continues to be a challenge. So, I think refinements of the technology is important. And then we still have to go back and see how can we harness the power of this platform potentially to hit cells that are actively proliferating, which might bring to bear the combination of AAV and gene editing technology. Mm-hmm. Yeah. So in our own work, we've been working on. This is an interesting twist in this. So we haven't talked about my little digression into leadership in the Gene Therapy Center at Florida, then in the chair of pediatrics, and now as provost and dean. But to talk about my own work, I'll just say that at a relatively advanced stage in my career, I've become more active as a trialist again, because some of our colleagues here at UMass, some of my good friends and colleagues who we helped to, or we did, I shouldn't say helped, we recruited here, Miguel Estevez, Bob Brown, and Heather Gray Edwards, they pushed because they wanted us to do a trial here for Tay-Sachs-Steinhoff disease, and said that they needed me to lead the trial because I am a pediatric subspecialist, and we've gotten into doing that, and that's become really rejuvenating for me in a lot of ways, and I'm really excited about that. And we are ourselves right up against these problems of global gene delivery to a human brain, even an infant human brain, it's a large, complex structure, and it's a daunting task even to engineer the right delivery approach. So we're back in the thick of that part right at the current time.
I think that this is fascinating. And I was just going to ask, I mean, you're wearing so many hats. I don't know how you do it. We'll get to this again, but I did want to switch over to your other hat that you just mentioned as well. You're the dean and provost of UMass Chan Medical School. I mean, what inspired you to move along that administrative path?
It's very interesting. Another kind of part of my career path is, I really started with running a lab like most of us, and we were growing in our impact at University of Florida. I was co-director with our dear friend who was recently passed away, Nick Musiska. I was co-director at a point in time with the Gene Therapy Center there. We were joined very shortly after I got there by Barry Byrne, who is the director there now. But we were moving things along quite well in the Gene Therapy Center. And at a certain point in time, I was asked to apply to be the chair of pediatrics. I had not had any significant administrative responsibility. Our Gene Therapy Center was really an amalgamation of colleagues who put in quite a few program project grants together and worked closely, but we really wasn't. We weren't doing heavy budgeting or any of that sort of stuff. But the reason I said yes to going back to a broader impact was really because of that what I'd seen and experienced and kind of envisioned at Johns Hopkins as a young pediatric resident. The fact that, a pediatrics department could help train people that would maybe be mostly taking care of patients, but would be in an environment where the frontiers of science were being pushed forward to try to provide solutions for patients who had conditions that we really couldn't treat effectively at the time. And I thought, wow, well, why shouldn't somebody who's oriented toward molecular medicine of the future really be, kind of the person to lead clinicians and researchers and educators, and then also, have those role, provide a role model for the learners. And I think we really got that department on that track. And then, becoming a provost and dean kind of evolved that out of that. And it's really kind of just an expansion beyond pediatrics. But, everything I've done in my individual career is based on trying to help patients, whether it is through research or through direct patient care. And really, that's what medical schools at their best are designed to do. But one of the things I learned is that to do that, you have to have these close-knit teams of people where you have clinicians and basic scientists working closely together. And that's kind of what we tried to do when I was in leadership, both at Florida and since I've been here at UMass. We've tried to bring together great groups of people that include both those who understand the patients and the diseases and those who understand the basic biology and the vectors and the viruses upon which they're based. I think it's been effective here. One of my biggest recruits, I mentioned Bob Brown in neurology, but one of the other key recruits that we recruited here to help me get started on this medical school was Guangping Gao, another former ASGCT president. And Guangping has been a tremendous partner and collaborator. And I highly value his partnership on an ongoing basis. And between he and myself, we I think helped to bring AAV here. UMass was already really on the map for the discovery of RNA interference by Craig Mello, which was just immediately preceded me coming here, was when he got the Nobel Prize in 2006. And so there's been a huge focus here on siRNA technology and gene editing now, but now we've kind of brought the gene therapy side together with that. But in many, many, many other ways, I think many other departments, we've tried to foster an environment where we can cultivate faculty all the way up, or I should say people really all the way up from when they're students, residents, fellows, young faculty, postdocs, so that they can use their talents. And my main job as dean is to recruit and retain the talented people here and help them to develop. And that's really what we've tried to do here.
I think that's been so critically important, and you have really done a wonderful job. I say critically important because it's obviously gotten also more difficult to get NIH funding, and so it's very important for especially junior investigators and junior faculty to get the institutional support to continue a career. And what advice would you give now younger researchers or investigators, really, when they come to you and looking for a fellowship or a faculty position? What do you tell them?
A few things I tell them. I think first you have to start with something you really love doing, that you're really passionate about, because it's no matter what you choose to do in academic medicine or science, it's going to be hard, and you're going to face many days of frustration and disappointment. So you have to have that burning desire in yourself to keep moving it forward. So you have to find something that you're passionate about, and then you can set a goal. Doesn't have to be that far off in the future. I never set a goal of being a dean, by the way, or even being a department chair for that matter. But my goal has been to try to come up with new treatments for patients that will really kind of have a major impact on them. So that's been my goal. But set that goal, but then make sure that you have both the time and resources in wherever you're choosing to be that will give you time to focus on that. I've become later in my career more broad, but that whole period of time when I was at NIH and as a junior faculty at Hopkins, when I first went to Florida, I was really focused. And then after I kind of really established myself, that's when I was able to get more broad. So focus, find something you're passionate about, focus, and then, just organize yourself in a way that you can have near-term and longer-term goals. Decide what your next two papers are going to be. And then the last thing I'll say for any scientists, really important, listen to your data. We have surprises all the time. I'm continually being humbled by what I thought I knew, and then what we find out we were wrong about. And you can go all the way back through all the history of AAV or all the history of different aspects of different diseases we've been involved with, and I think realizing we still have more to learn than what we know. It's a hard thing to do, because sometimes when somebody gets an unexpected result, they view that as a big disappointment. But as long as you've done the right controls, right, it can be a really important observation. Anyway, I'm sure you know that.
I know that. The only thing I'd add is resilience. So
all of that takes resilience, right?
So Terry,
disappointments.
If you had a crystal ball, right? So what do you think we're going to be in 10 years with all this gene therapy and gene editing in particular now? Any thoughts? Or where would you want us to be in 10 years?
Yeah. Well, there are two slightly different things. Okay. So where I think we're going to be, I certainly think we're going to have scores of gene therapies approved, in the United States and Europe and other developed countries, and maybe, some low and middle income countries even. The fact is that the technologies that we have now do work and work at a level sufficient to treat many different diseases. And I think just as we see the proliferation of monoclonal antibodies now, we'll see a parallel proliferation of gene therapies for specific conditions. And those will include various ones, including not just AAV, of course, lentivirus vectors, gene editing, technology delivered in various modalities, ex vivo, as it's already blossoming in immuno-oncology in various areas. They'll be spread across the areas that we would see represented at the ASGCT annual meeting. I'm really hopeful that we'll begin to crack the nut on a few other higher prevalence conditions. One thing I didn't mention is my mother had died of Alzheimer's disease.
Yes.
Terrible condition.
Yeah.
We've got vectors that get genes into the brain reasonably well, and if we knew exactly what to deliver, that might be a terrific one to see fall into the category of somewhat treatable.
Yeah.
Now, along the same lines, so you asked what I would want to see. Well, I'm going to relate back. I did not choose when I was a pediatrician at Hopkins or pediatric resident at Hopkins. I did not choose to do pediatric genetics, even though I've worked on genetic diseases. And one of the reasons was somebody told me, and they at the time I think were right, that when you work on genetics, you can learn a lot about a disease, but you can't do anything about it.
Right. Yes.
That was told to me like in 1986 or 87, so it was true in 1986 or 87. But where I sit right now, because we are involved in pediatric genetic diseases, is that families are contacting us all the time and saying, I have this other genetic disease. I see what's happened for SMA1, what has come forward and has been approved. Why can't we use this for my child's disease?
Mm-hmm. Right.
And of course, the ASGCT is very good at pointing this out. I mean, there's somewhere between 7,000 and 10,000 rare diseases or rare genetic diseases. And I would love to see us be able to continue to spread out across many, many more of these ultra rare genetic conditions, because this population of people really, they are people we have used as a scientific community to learn about genetics and to learn about molecular pathways. And I think we owe it to them to put some of that science back into those communities. So it continues to be a disappointment. For some conditions, we can say there's a treatment. For others, it's still not here yet.
Yeah, no, I totally agree, Terry. One last question I have for you. What do you do in your free time? Is there any free time with so many hats you're wearing?
Well, several things. We're sports fans. My wife and I have season tickets to the Woo Sox, which is the Worcester Red Sox, the Triple H franchise of the Red Sox. This year, I think the Woo Sox may actually end up being better than their Boston colleagues. But I don't want to get hate mail from them. And the other thing I like is I have to confess that I love obscure sci-fi. I will sometimes sequester myself with the remote, and also I do occasionally kind of look at unusual, divergent foreign language films on the Criterion network, and I like kind of weird dystopian future types of movies. I don't know why, but I don't like superhero movies, but I like sci-fi movies. So there you go.
Okay. Oh, thank you so much. I didn't know that. Yeah. Thank you so much, Terry. I really appreciate it. And thanks for all you do. Thank you.
Well, thank you so much, Hans-Peter. You've been a great president for our society. And, I love seeing you with your, I don't know if other people are going to be able to see your background. I love seeing your background with Seattle and Mount Rainier, because that is the birthplace of the ASGCT.
That's right. Yes.
We didn't know that.
Yep.
So I've fond memories of times I've visited Seattle, and love to visit it again soon, hopefully for future ASGCTs at different times.
Yes, I hope so. Okay. Really appreciate it, Terry.
All right. Take care.